Hi everyone, I'm Mark Braceland. I'm the Director of Fish Health here at CAT, and I want to talk about emerging diseases in aquaculture. So emerging diseases, or I guess we could reclassify that to emerging infectious diseases, are typically defined as infections or diseases which are increasing in outbreaks, presence or severity, with newer altered pathogenic agents in a new host or region. And we can also extend that as well to encompass diseases or infections where mitigating strategies have reduced in efficacy in recent times. And they pose a high threat to our sustainability in fish in, fish out ratios and production, primarily due to our lack of understanding and knowledge of them. So there's several examples of these globally in Atlantic salmon production. And really, it seems that we're on somewhat of a 2-year cycle with emerging diseases where we have new pathogens and reductions in treatment efficacies coming up in geographical limited locations and then also globally. So we have Pasteurella skyensis, for instance, also several gill pathology diseases, Salmonid rickettsial syndrome, and then ISA remains a perpetual threat to our sustainability as an industry. Today, I'm just going to focus on our experiences with two of these kind of emerging diseases, one being Tenacibaculosis and then also infectious pancreatic necrosis, which with some recent reports seems quite topical of potentially a re-emergence in that disease. So Tenacibaculosis was first described in BC in Canada on the west coast in 1988 by Kent et al. And subsequently in other regions. So in Norway, first being described in 2010. And since that first description, instances of outbreaks and severity of disease and thus losses due to Tenacibaculosis have increased steadily. And then in the last 5 or so years, we are seeing an exponential increase in losses due to disease. Tenacibaculosis is caused by primarily three, well, in Atlantic salmon, it's caused primarily by three species, so Maritinum, Finmarkensi, and Dicentraki. And it's interesting in that often Tenacibaculosis can be caused by either a single infection by one of these isolates or species, but we also see often complex Tenacibaculosis where all three of these species can be present on an Atlantic salmon production site at a given time. So to also note that other salmonid species have been found to be infected and they can cause disease in those species. So in 2020, there was a first description of rainbow trout and coho salmon being infected and pathogenesis ensued from that outbreak in Chile. And there's several well understood or increasingly better understanding of several of the risk factors associated, such as time to post-smolt, temperature and season, and also salinity. Our initial work focused on 12 Tenacibaculum isolates from BC, and we were able to identify quite quickly that we can kill a lot of fish very quickly with several of these isolates. But through our work, we tried to understand a number of the influences of virulence on the bacteria itself, fish susceptibility, and then also the gross pathological manifestations. So through our work, we tailored things in terms of the raising of the bacteria colonies, in terms of dosing and dilutions, and also the effect that environmental conditions can have on how these bacteria grow, and thus the impacts that it has on virulence and resulting mortality. Also in the fish as well, understanding the effects of survival due to time post-smolt, and then also environmental conditions at the time of challenge and during challenge. We've been able to successfully develop a couple of nice Maritinum models that would be useful for further work and development of new technologies and mitigation strategies. This is just one example of some work we've been doing. This is in collaboration with Sbtype, where we were able to demonstrate the significant effects in increasing survival that we can have through functional feeding. Also just want to highlight some of the difficulties that we have in diagnosing Tenacibaculosis in the field, and also understanding the causative agent, both in terms of species and then in subtypes of a given species. So currently there's over 25 species of Tenacibaculum that is documented, not all of them which have been shown to be infective or disease agents in Atlantic salmon. But then even within a given species, there are globally over 100 subtypes identified in Tenacibaculum martyrum. And also available qPCR tests are unreliable in identification of a specific species. And that's especially the case in our experience with Finnmarkensi and Dicentrachia. They are genetically very similar, and thus qPCR methods are quite limited in being able to tell those two species apart. I think that's also compounded by the effects of mixed infection as well, in that, you know, some pre-analytical methodologies can also have a big influence on our ability to identify specific species and subtypes. We recently did some work with a phenomenal summer student called Ian Douglas, who joined us earlier in the year, and potentially have identified a new Tenacibaculum species from the west coast in BC that had previously been typed as a Finnmarkensi, but through the sequencing of 7 loci, we were found it to be an outgroup, and we're currently trying to investigate this more, both in the wet lab, trying to see if it's a disease-causing agent, and also through whole genome sequencing, hopefully can identify if this is indeed a new species of Tenacibaculum. So just to quickly summarize our work thus far with Tenacibaculosis, we've developed a Tenacibaculum martyrum model, which can aid in the development of novel technologies and mitigation strategies against Tenacibaculosis. The current abilities of us to identify species and isolates in different regions is limited due to the qPCR methodologies that are currently in the literature and commercially available. We're also extending our work in the wet lab to other species and isolates from different geographical locations, with a high interest in understanding the differences in close presence and severity or virulence markers of isolates from Chile and Norway and Scotland as well, to add to the work that we've done thus far in Canadian isolates. I think also an important step for our understanding as an industry in Tenacibaculosis is that of the development of a complex Tenacibaculosis model that can be available to the industry, because we know that outbreaks are often not of a sole subtype or species. Conversely, infectious pancreatic necrosis is a well documented and understood disease of Atlantic salmon. It's the first virus that was ever in vitro isolated in cell types in 1960, and then first identified in Norway in 1975. Some 25 years after its first description in Canada. We've also, as an industry, benefited greatly from the understanding of resistant QTL, and since implementation of that technology, outbreaks have reduced by over 80% throughout the industry. However, there has been some recent reports of new virus variants and potentially outbreaks in QTL resistant fish. I just want to talk about some of the experiences we have had with IPNV and some of the drivers of both virulence and host susceptibility. So one of the first studies that we did with IPNV looked to understand the effect of both host virus, and then also of environment on mortality that's induced. So in this study, we looked at 5 different strains or sources of fish, 3 from Norway, 1 from West Coast Canadian strain of fish, and then also an East Coast strain or species of Atlantic salmon, which is St. John River subtype, which is endemic to Eastern North America. We did this 5x3x2 design using 3 SP serotype isolates from Scotland, Norway, and then Canada, West Coast Canada, and then looked at the mortality that was induced via cohabitation experiments in fresh and salt water. Firstly, what I'd say is that we found that we had low mortality in all fish strains in fresh water, and we also had very low levels of mortality when we used the Norwegian and Canadian viral isolates. However, we did see some interesting results in strain differences when we used this Scottish isolate in salt water. This isolate originates from Shetland and was first isolated from an outbreak on a sea farm in 1999. So that led us down to try and understand why we saw this difference in fish strain and source. And one of the first things we investigated was the effect of just size at the time of challenge. And we found that that held very well in the Norwegian fish across those strains of European stocks that we tested, where we found that the smaller the fish were at the time of challenge, we had higher levels of mortality, although it should be noted that the fish did smolt well and there was chloride tests done, however, there could be some physiological effects that cause them to have higher susceptibility and thus greater levels of mortality. So as we further developed this and investigated why we had this differential survival across fish strains, we looked at a subgroup of fish from these different strains and typed them for a non-synonymous mutation in the epithelial cadherin 1 gene or loci that was published by Moen in 2015. Our sample size for the European stocks or Norwegian strains was quite low and didn't really allow us to have a clear picture. However, with the St. John strains, there were some interesting results that were obtained. In essence, what we found was that in the St. John strain fish that we have tested thus far, that there was a very low number of homozygous resistant SNPs or CC genotype. There's actually only 3 in the whole population that we tested, although despite this, there was only 2 mortalities and 1 survivor. And when we've tested St. John River strain as mixed genotype cohabs and then Moen SNP associated susceptible genotype cohabs, we found no significant differences between studies. Well, there has been some differences. We've also done a lot of work trying to understand, when we go to do wet lab trials, we have to propagate up virus enough to infect frozen fish for introduction to naive populations. And this is quite an interesting study that we carried out, understanding the effects that cell passage has on the virulence of the viral isolate. So we grow our IPNV on CHSE cells. And these 3 mortality graphs or survival curves show the differences that we induce when we have 1, 3, or 5 cell passes. So this reduction in virulence is well described and can be identified via a single point mutation in the VP2 region at 0.221, where we often see a substitution of an alanine to a threonine. However, while this threonine SNP is well characterized and holds up throughout the literature and also in our own work that we've done in predicting a low virulence in fish, there's several conflicting markers of virulence in viral isolates. And indeed, our own isolate is quite different. We often, our isolate has a serine here at 0.221 on the VP2 region, or loci, sorry. And this is not well classified. We've only found one other paper, Marquez et al. 2016, that's found a serine mutation at this point. And that paper identified this at this point from an outbreak in Chile. We're still early in these kind of investigations, and potentially there's an effect of St. John River strain, but this relationship of this Moen SNP and this serine certainly warrants some further investigation. So just to conclude on this, the work that we've done thus far, I think that there's a lot more investigation required throughout the industry on the effects of new subtypes and variants of IPNV and what that may mean for us in the industry. In our work thus far, we've identified that the predictive nature of this Moen SNP is in epithelial Keterin 1 is also predictive in St. John River strain. The influences of the virus itself and markers of virulence in this is warrants further investigation. Also, it highlights the importance of sequencing straight from fish prior to passing or doing kind of traditional viral isolation in cells, as the virus is readily changeable, and these SNPs can have big impacts on predictions of virulence of the outbreak itself, and also can impact significantly in vivo model development. So lastly, I really want to first of all thank an amazing fish health team. There are many of us now. There are 17 in total. A couple of camera shy people didn't want to be part of this photograph. I want to thank them for all of their work and dedication that they have for us as a team and a company, and for our clients as well. Without all of these people in this photo and some others that aren't in that photo, the work that we do would not be possible. Also really want to thank Marine Scotland, specifically Ian Munro, for sharing with us a number of isolates, including this IPNV isolate that was discussed. I also want to thank Moen in Greek Seafood in BC for the provision of far too many Tenacibaculum isolates that I can count. And I want to thank you all for your attention, but with the cameras being off, I don't know how many people are asleep, but I'm going to presume most of you are awake, and thank you very much. Cheers.